Bismillahirrahmanirrahim. Today I would like to share the knowledge about Leishmania. My name is Sarfraz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share the knowledge with references these books. Veterinary Parasitology 2nd edition by G.M. Arcohart, Veterinary Parasitology 4th edition by M.A. Taylor, George's Parasitology for Veterinarians 10th edition by D. White, D. Bowman, and last one is the internet sources. Please subscribe my channel and also push the button of bell icon. The class Mastigophora. The genera is the most important six genera in the class Mastigophora. The Mastigophora have the following six genera: Trypanosoma, Leishmania, Trichomonas, Histomonas, Hexameta, Giardia. So now the Leishmania is derived from the name of the William B. Leishman, 1856 to 1926, a British pathologist. What is the Leishmania? It is a single-celled parasitic protozoan, and they spend a part of their life cycle in the gut of a sandfly, and they also spend some portion of the life cycle in the blood and other tissues of a vertebrate. And the disease is called Leishmaniasis. This is the vector-borne disease. Leishmania are the final host man, dog, and a wide variety of wild animals. Intermediate host, blood-sucking phlebotomus, sandflies. Site of infection that the protozoa multiplying into the macrophages. And a species, the Leishmania tropica, causes the cutaneous leishmaniasis, and it is also called the oriental sore in the old world, the eastern hemisphere. The Leishmania donovani causes a systemic or visceral leishmaniasis. It is also called the Kala-Azar, and its origin is from the Calcutta in India, the first time reported, and it's also the name. Other name is the black fever or the dum-dum fever. Leishmania brasiliensis. The causes the lesions similar to Leishmania tropica, or is also called mucocutaneous leishmaniasis, and distribution southern Europe. The life cycle of Leishmania. First of all, I would like to tell the preparation period or the life cycle, that the 4 to 17 days, and incubation period is 2 to 4 months. So this life cycle is divided into two main parts. One part is that the extracellular life cycle, and other one is the intracellular life cycle. This extracellular life cycle is take place in the sandflies, while the intracellular life cycle take place in the host mammals. So in the sandfly, this non-infectious promastigote changes into the infective promastigote, that is the metacyclic, and it is also called the infective stage. This is the infective stage of the Leishmania, that is the promastigote. This promastigote, when the sandfly bite, and it is engulfed by the macrophages of the promastigote, and this macrophages promastigote, in the macrophages, this promastigote is divided into change into the amastigote. This macrophages amastigote, when the sandfly bite and it sucks this amastigote, and this amastigote in the sandfly then changes into the non-infectious promastigote. So this is the life cycle, and I told you that the two main parts. So the difference between the types of Leishmania, first one is the cutaneous leishmaniasis. This is also called the dermal leishmaniasis. This caused due to the Leishmania major, Leishmania amozensis, Leishmania infantum, Leishmania donovani. These, when the sandfly bite, it make the macules at the site of inoculation by the bite of infected fly, and this macules changes into papules. Then papules are the painless ulcers and lesions at the whole body. And old world, the eastern hemisphere, it is called the oriental sore, single and multiple ulcers, while in new world, the western hemisphere, this is the Calistro ulcer, and it is the single and multiple ulcer. So another type is the mucocutaneous leishmaniasis. This is also called the mucosal leishmaniasis or nasopharyngeal leishmaniasis, the third clinical form, and a severe erosion of nasopharynx, facial collapse, destruction of oral and nasal tissues. So the third one is the visceral leishmaniasis, and this origin is Kolkata in India. First time reported Kala-Azar. It is also called the Kala-Azar, black fever, dum-dum fever, and its species is the Leishmania donovani. Parasite inoculate by the sand fly, the bloodstream, and chronic fever, enlargement of the lymph nodes, enlargement of spleen or liver. So deficiencies of red blood cells, white blood cells, platelets, and it causes the anemia, weakness, subclinical infection, and in IP period, infectious incubation period is the 2 to 4 months. Pathogenesis activated proliferating macrophages, chronic enlargement of the spleen, liver, and lymph nodes, hemolytic anemia. So the clinical signs: shallow skin ulcers, generalized loss of body hair, eczema, skin become that is the inflamed or irritated, fever, anemia, myocarditis, heart failure. So the epidemiology found in people in focal area of approximately 90 countries in the tropics and subtropics and southern Europe, and more common in rural than in urban areas, but it is found in outskirts of the some cities. Ecological that settings range from rainforest to deserts. So diagnosis, clinical diagnosis, then the thick smear or maybe GIMS or Leishmania stain, and then PCR. So the treatment, antimony all compounds, sodium stibogluconate, 20 milligram per kg body weight for 30 days, and appear to inhibit the bioenergetic pathways such as the glycolysis and fatty acid oxidation in Leishmania amastigote. Another is the allopurinol. It's in the market the zaloprime or alloprime, and the potassium antimony tartrate, and 100 milligram per day for adult or oral or intravenous. Its mode of action is that inhibit the xanthine oxidase, an enzyme in the that is protein the catabolism pathway that converts the xanthine to uric acid. The control the destruction or killing of the infected dogs or stray dogs, because these are the reservoir host. So thank you so much.